Good morning. It's a pleasure and an honor for me to welcome you all to this EFSA Scientific Colloquium number 25. The title is A Coordinated Approach to Assess Human Health Risk of Micro and Nanoplastics in Food. So, my name is Helle Knudsen and I'm chairing this colloquium. My work is at the Norwegian Institute of Public Health as a scientist. And I'm also a member of the EFSA NDA panel, the panel on nutrients, novel foods, and food allergens. But I was previously the chair of the content panel, and that was the case in 2016 when this panel was behind the EFSA statement on micro and nanoplastics in food. Well, I think that's enough about me. I should also say that I'm lucky to have Tobin Robinson with me as co-chair, and he is the head of EFSA Scientific Committee for Emerging Risk Unit. As you all know, we should have been in Lisbon last year, and we all know why that didn't happen. And it's the same reason we are not in Lisbon now as we should have been. But this is how it is. The registration was full and closed already more than a year ago. And probably even more people would have liked to attend this event. But luckily, it is live streamed, so more people have the chance to follow. And you can, in fact, send us messages on Twitter and with hashtag Microplastics21 if you want. We have 147 registered attendees. 85 of you are from EU countries, but 36 are from non-EU countries, and we are, in fact, from 5 different continents. Most of us are from universities and research institutes, but we also have many participants, 40 from national authorities. It was 51 from universities. And 32 of you are coming from the private sector. And the rest includes EFSA staff, panel members, NGO representatives, international organizations, and also the press. So I think altogether we are a good mix of people, and which is exactly what we need for this colloquium. So since we are not physically together in Lisbon, we just had to imagine that we are sitting in the same room. And I hope that we together can make our imaginary room so that we can have a fruitful discussion and contribute to moving this field forward. We have an animation that I think capture most of the aspects that we are going to discuss over the coming 2 days.
Microplastics are tiny fragments of plastic. Nanoplastics are even smaller, and they are invisible to the naked eye. Some of these tiny particles may be produced this size, but often they are fragments of larger pieces of plastic, which over time break down and disperse in the environment. These plastic particles vary in composition, quantity, quality, shape and size. Scientists need to standardize how to identify and quantify these particles to generate data on their occurrence in food and on any harmful effects on people, both from the particles themselves and from any chemical or biological contaminants they might absorb.
And by this, I give the floor to Barbara Gardani. She's head of EFSA's Communication, Engagement and Cooperation Department, and she also wants to wish you welcome.
Thank you very much, Helle. And we have different backgrounds. We're in different rooms, but very much in the same space. So a very warm welcome to all participants in this 25th EFSA Scientific Colloquium. The issue of microplastics in the context of food safety was first brought to EFSA's attention in 2013 in the context of the Emerging Risks Exchange Network. And in 2015, we were asked by the German Federal Institute for Risk Assessment, BFR, to provide a statement on the presence of micro and nanoplastics in food, with a particular focus on seafood. And the terms of reference for this work covered the potential hazards to human health, data gaps, and research recommendations. So EFSA Contam panel, and Helle was working as part of that, issued a statement in 2016 with recommendations in 3 areas. And these 3 areas you will recognize as part of the structure for the colloquium today and tomorrow. The areas were analytical methods, exposure assessment, and hazard identification and characterization. And there are still 5 years on quite a lot of questions open, and it's very exciting to be able to collate so many experts from many different countries within and outside the EU to really address these questions together. Now, the importance for the EU of micro and nanoplastics is evident. Plastic pollution was highlighted as a priority in the European strategy for plastics in the circular economy and bioeconomy strategy, with a focus on plastic recyclability and plastic-free environment. And in the Horizon 2020 research program, a research call was launched in 2019 to follow up on the EFSA recommendations from 2016. So there's still a lot of ongoing work. And recent reports on the topic have been published by many institutions, including SAPER, the Science Advice for Policy by the EU Academies, which is part of the scientific advice mechanism, by the Food and Agriculture Organization, by the European Chemicals Agency, the European Environment Agency, the Joint Research Centre, the World Health Organization. So there are so many of us working on micro and nanoplastics, and it's great to have the opportunity to get our heads together to continue to resolve issues. Now, this is a very complex scientific issue. It's peppered with uncertainties, with data gaps, and it's clearly work in progress. And these are characteristics that make science difficult to communicate. And this can generate some consumer concerns. In 2019, EFSA's Eurobarometer on food safety confirmed some of the findings by the BFR Consumer Monitor. What I mean is microplastics in food is a topic of concern amongst EU consumers. And in the news, we see nanoplastics in drinking water, in seafood, in honey, in snow, in ice. It's quite a wide range of really fascinating news pieces that are shaping the public discourse and the opinion. So the aims of the colloquium are to foster discussion, collaboration and knowledge sharing, to facilitate food and feed safety risk assessment, to set priorities and challenges for policy making related to microplastics, to review the available research and identify research gaps since the opinion of 2016. And based on what I said about the science communication challenges, we also wanted to introduce in the colloquium a discussion on social and behavioral insights in the area of micro and nanoplastics. As I said, the breakout sessions, so the structure of the colloquium is going to be around the 3 areas of the EFSA's 2016 statement: analytical methods, exposure of humans, hazard identification and characterization. And all the work and all the ideas and the exchanges that will happen between today and tomorrow will be captured and published in a special issue of the EFSA journal. So what is left for me to do in this short introduction is to ask you to consider and to answer a poll, which you will see on the screen. And the question is, we are here to discuss a coordinated approach to assessing the human health risks of micro and nanoplastics in food. And what do you think are the main challenges for us at this point in time? A, produce more evidence. B, better coordinate existing research. And C, engage scientists in more discussions with consumers and with the general public. So I would like you to spend the next 30 seconds thinking about what you think is the priority amongst the three on the screen. And I will hand over to Helle and wish you all a very engaging and a very productive couple of days. And I'll see you again, most probably tomorrow, if not before. Have a good colloquium, and back to Helle.
So, while we are waiting for the result of the poll, I think I can say some words about the plan for today and tomorrow. So, today we will first have 3 talks, 30 minutes each, and including questions. And we will have questions after each of the talks. Then we will have a coffee break for 20 minutes, and after the break, 3 different sessions. So it will be a development in analytical methods as session 1 before lunch, and after lunch we will have exposure and in the end hazard identification and characterization. Tomorrow we will start the day with the breakout sessions in 6 different groups covering the 3 different topics. And you will receive more information on that before we end today. And I think the poll is getting ready. Okay, but after the break tomorrow, we will gather in the plenary again, and we will have short reports back from the breakout groups, a plenary discussion, a panel discussion, and then wrap up and closure. And the plan is to finish at 1 p.m. As I said, we will have questions between each of the talks to the extent that time allows, and you will have to submit your question in the chat. Please indicate who the question is meant for. It's probably to the speaker, and I will read the questions for you. You can also control the screen via the menu, or you can control the screen via the menu. And that's on the bottom of the screen, and it should appear when you move your cursor. And there you also have the chat. Finally, I remind you that you have received a link to the microsite and a practical guide. But I should also say that you have the electronic posters and that you will be able to book a call with the authors if you want to. To enter the poster session, you have to follow the link that you received the 5th of May. So we have the results of the poll. It seems that it's to engage scientists in the media is no, it's the no answer that is the largest. Sorry. Better coordinating the existing research. So I think that's got the highest score, followed by produce more evidence. So we'll see what we think of this tomorrow. I think it's time to hand over the floor to the first speaker. And the title of the first talk is Micro and Nanoplastics and Human Health. And this talk is given by Bart Koolmans at the Wageningen University Research. So please, Bart, you have the floor.
Thank you, Helle, for your kind introduction. I was asked to provide an introduction, a very global one, and global is also what you probably feel popping up in your mind looking at this first slide. Plastic pollution, microplastic pollution, nanoplastic pollution being a global issue. I think that's clear. So in this introduction, I will touch upon many issues, so it will not be a very deep overview, but a lot of things I will briefly mention and introduce. There will be a brief history of what we have learned about microplastic, nanoplastic, and human health. And I will try to provide a brief overview of where we are, the state of the science, but most of the times ending with a question rather than ending with the answer. The answers will be given later, I guess, in other talks and discussions. So what do you see here is an axis of risk, and this would represent the more technical risk. So risk, as we would see it, as natural sciences. And and then we do not know a lot about this technical risk, but still many experts would probably say that especially for the larger particles, the body of evidence for risk is fairly low. We are a bit more at the left-hand side. Still, this is surrounded by considerable uncertainty. There is another axis, and that's the level of concern, and it was also briefly mentioned a couple of minutes ago in the introduction. The level of concern is different. The mechanisms are different. So you might think that why are we not here? The level of risk, which is really demonstrated in the literature or in formal assessments, might make us think that, the concern, how does that work? Now, actually we're here, and of course then the logical question is, what explains the difference? What are the mechanisms behind this? And that has to do a lot with how perceptions work, how technical risk works, how scientists communicate about these technical risks, how scientists as well as the public perceive risks. I will not provide the answer about this, but there will be a presentation later on, for instance, by Sabine Paul, who will provide a more refined view on these mechanisms, and hopefully will reveal a bit more about this, what's in this question mark over here. Then my next topic for you is a brief history of major reports, and not everything is here. So there are more important reports and papers. This is just a selection of a couple of reports that you probably know or have seen. So in 2016, the EFSA report, 2017, the FAO report, 2019, there was an expert report by SAPIA, WHO report about drinking water specifically, 2019, and there was the first quantitative assessment of what people consume, Cox et al., the World Wildlife Fund report 2019, and very recently the first probabilistic assessment of exposure in a recent paper in ESNT. Now, at the left-hand side, lower in this slide, you see that all these reports provide a further refinement of the estimates with respect to exposure, but they all say, even if we have further refinement, it's still far from complete. They also all say that there is not much evidence with respect to hazard, but that does not mean that there is no hazard. It is not demonstrated yet, but especially what they say is that more research is needed. And yeah, I guess that makes a lot of sense. Nanoplastic. What do we know about nanoplastic? This is just an example I took from the SAPIA report, where we provided different colors for different levels of knowledge. So there were things that we knew, there were things that we were a bit less certain about, and there were things that we said, we do not know anything. And basically nanoplastic was in that gray zone. We did not know a lot. And that is still the case. So Occurrence, sampling and analytical methods for nanoplastic is not yet established. For drinking water and food, which is important for the symposium, nanoplastic in drinking water, there's no information at all. Exposure, wastewaters, nanoplastics are unknown. We think they are coming from larger plastics by fragmentation and so on, but we cannot be sure. The mechanisms are still unknown. And then, of course, the key question of risk. If you do not have these earlier aspects like occurrence data and exposure data, you cannot assess risk. So no risk assessments have been published for nanoplastics. The occurrence and risks of nanoplastics are, you could say, a highly plausible but still formal hypothetical risk. Another thing that we will probably talk about later in this colloquium is the uncertainty due to the diversity of microplastic, nanoplastic as a material, symbolized here in the left-hand side upper corner. Many shapes, sizes, dimensions, so that is clear. Now, if you look at this graph, these curves that you see then for solutes, there is uncertainty in effects and there's uncertainty in exposure. And for a characterization of risk, this would propagate into a certain overall uncertainty. And now the problem is that due to this diversity of microplastic and nanoplastic as a material, you could think of it as the same. However, these uncertainties are much larger. The distributions are much wider because we're talking about a suite of contaminants, you could say. And from the paper that I mentioned already, this is an example illustrating that. So this is a graph where you see exposure, and on the x-axis it is milligram per person per week. Now, what the median is, that's clear, the 50% percentile, where most of us on a median basis could be exposed to if this paper would provide the correct data. So that is a big group that large group of individuals. But the uncertainty is huge. It spans from 10 to the power of -6 to 10 to the power of +6. So it also illustrates, hopefully, that reporting a median is tricky, because the chance that the median will be correct is just very small. It might be very useful to just quantify these distributions. Then this scheme here, which looks very, very complex, and I guess it is a scheme that I will use in a couple of slides now to explain some of the mechanisms concerning exposure and effects. So at the left-hand side here, you see a flow of information that you would need to address if you want to understand exposure and uptake of plastic. So this is a plastic model. At the right-hand side, a similar workflow if you would like to address what chemicals associated with plastic do in terms of exposure. That recent paper that I already mentioned provided estimates of exposure, and that's here plotted vertically, and you see these violin plots. And this is a comparison with empirical data, microplastic concentrations in human stool, and that's the dashed line. And the yellow violin plots, they correspond, that's the model prediction for that line. So from this we would learn that the empirical data, it's limited data, it's only one study actually, but still it is not inconsistent with the model prediction, but the model underestimates by a factor of 7. Now, however, this model also accounted for only 20% of the diet by weight, so that it means that this at present is not inconsistent. We need more data, we need refinement of the models, but at least at this stage, this is not inconsistent. This model, just going on with representing a bit of the state of the art in what is done in the literature, this model also predicts what's absorbed by the body. A lot of assumptions like 1 to 10 micrometer being absorbed, 0.3% considered to be absorbable for different scenarios. So there are tools available to further detail exposure estimates, but it's still highly uncertain. For instance, this only represents 20% of the diet components by weight. What about the other 80%? So basically, we do not yet know enough about what is in different diet components. Another issue is that whatever is measured in drinking water thus far, or some of the other food sources for which we have information, it's done with different methods, targeting different size ranges. So data are often incomparable. Another problem is what exactly happens in the gut in terms of absorption and rates and distribution parameters. So largely unknown, actually. Another aspect where we gained a lot, I think, is recognizing that quality assurance and quality criteria are very important. There is a lot of value in almost any study that has been published, but when this information needs to be used for more formal risk assessments, for instance, in a regulatory context, and I guess that's what we're heading at one way or the other, then data should be fit for purpose. And it is good to see that some systems have been developed that was not inventing the wheel, because this was simply building on earlier similar systems for ecological or medical studies. But there are tools where you can take a look at the data in the literature and then take some criteria that have been formulated with respect to the technical quality of a study or the relevance or fit for purpose aspects of a study. And then by combining all these data, you can get some view on how useful is this data set for more formal risk assessment. And it provides you overviews like this colored bar graph overviews of in what respect studies provide sufficient information for risk assessment. So then about the chemicals. I have a quick note in between for the organizers. My clock started at 20 minutes where I thought I had 30. So either please correct me if I'm wrong, or otherwise I will just simply speed up. So for chemicals associated with plastic is also a very important aspect. And just an overview of different outcomes of reports here at the right-hand side plotted. So the EFSA report looked at this and they used a worst-case approach, and they concluded the effect of plastic-associated chemicals is very small. FAO, similar approach, also very small. Sapir report reviewed the evidence and concluded that it's likely that the effect of plastic-associated chemicals will be small. WHO the same, and also in the recent paper by Mohammed Noor, where this was not done in the worst-case approach, but probabilistically it was very small. The conclusion from these reports also is that it is context dependent, but assessment tools are available. So in this red circle over here, there is a way of modeling modeling the processes that occur in the gut that you just can use to exactly calculate what happens with chemicals that are associated with ingested plastic. A brief outline of this here. So this is a bit more in depth, I guess, but there is a framework available to assess the chemical bioavailability of chemicals associated with plastic in the human gut. And this starts with empirical data. So you see a brief overview of empirical results. At the left-hand side here, you see basically binding constants. So the affinities of chemicals in the gut for gut components, like the micelles and the food that's available and the lipids that are present in the gut coming from ingested food. So this is more like, I read environmental chemistry, like binding constants and hydrophobicities and all that. Then in the third graph, you see that's the kinetics. That is how fast microplastic particles take up chemicals from the gut. So if the gut is contaminated due to contaminated food, a clean or cleaner microplastic particle will pick up the chemicals. And you see that this is a fast process. But it also can be the other way around. In the fourth panel here, if the plastic is contaminated and the gut is clean or cleaner, then you will see rapid desorption. And the last one here is also interesting because that's when the plastic particle and the food both is contaminated in a similar way, and you still see an increase according to this empirical data. And that's because the food is digested, and then the chemicals on the food are released or liberated, and they are captured by the plastic and then leave the gut. These last three panels represent different scenarios. So what happens? And that is the context dependency that I was already mentioning. It depends per chemical, per food source, fugacity gradients and all that drive this. But it's very relevant to realize that this can be captured in a model. A model is available. So you would say there's chemicals in the gut fluid, on the plastic, on the micelles, on the food. Food is digested, and this can all be kinetically modeled. So with some mathematical equations, we would get a set of parameter values. And then if you have all those parameter values for a whole suite of chemicals, you can use this for scenario studies and basically just calculate for any situation what the extra exposure will be due to microplastic ingestion from a food source, or reduced exposure due to a certain specific combination of plastic and food source chemical properties. All right. This was all about exposure of plastics, exposure of plastic-associated chemicals. A few notes on hazard, and you will see that there's less because less is known. There is a lot done with respect to hazards and effects, but we are still a bit in a gray zone when it comes to understanding how what we know about hazards translates to actual effects and risks in an in vivo situation. I provide a brief overview of what I thought are some of the major challenges. And I know that in later presentations today, this will be more presented with more detail. So number one here is the uncertainty around how to translate observed effects in vitro and in vivo animal models. How can we translate that to in vivo scenarios? Many of the tests and studies done use particles, and we learn a lot from that. But are they representative for the particles that we are exposed to? And the smaller the particles get, the more serious this challenge could be, because for nanoplastic, we do not know exactly how it looks like. Yes, we know it's small, but the experiments are done with polystyrene spherical most of the times, and we do not know how irregular or symmetrical the actual nanoparticles are that we are facing in the environment or in our diet. Then there is a lot of uncertainty due to QA/QC issues with data. So to what extent can we apply them already? Then there is unknown bioavailability of plastic particles. So which fraction actually passes the gut and which fraction can be absorbed? And if it's absorbed, then which fraction gets stuck rather quickly and which fraction travels further? So to understand this and to get PBPK, the biodistribution kinetics parameters, requires a lot more work. And then number 6 here, microplastic particles will have some kind of hazard profile, some toxicological profile. But how does that compare to that of other particles? There is recently an approach which might be helpful to deal with the diversity of microplastics in terms of toxicological effects. represented here, that microplastic particles and the diversity can be captured by presenting them as distributions. So if we would know the distributions for length, width, and volume, and width to length ratio, and so on, or area, and we would have the distribution of these metrics for drinking water and for food, for instance, that would be helpful, because all of these somehow may relate to a certain effect mechanism. So what you see here, right-hand side lower corner, is the blue arrow, the upward arrow is basically representing that if there would be a toxicological effect mechanism, then we would need the toxicologically relevant metric for that mechanism. And then we get stuck, because how do we know this for such a diverse material? But if we can use these PDFs, probability density functions, these distributions like the upper part of this slide, if we could use that and combine that, that may be a way forward to quantify toxicological effects for such a diverse material. And then coming back to this aspect of, let's not forget the other particle types. So polymers versus other particles. Why is that important? Well, of course, we're interested in risks of micro and nanoplastics, but they are not coming alone. They might have effect mechanisms that they share with other particles. In this plot, you see vertically, it's the persistence of different particle types, and horizontally plotted, it's the size. Now, this is not the end of the story, but it is likely that things like persistence and size are important drivers of toxicity. The microplastic area here is the red contour, but you can see that, for instance, black carbon particles or soot particles, they also are very persistent, and they can even get smaller sizes, very small. Well, no need to detail this entire slide, but there is similarity and dissimilarity with different particle types, and at some point it might be useful to consider that. And then this is my final slide. In summary, when you just Google microplastic in human health, you get a lot of questions and subtopics and concerns, which brings me to my final introductory comment. There's a lot of work to be done. Thank you for your attention.
Thanks a lot, Bart. I think we have a very shy audience because we have no questions that have been posted in the chat. So actually you have time if there are things you would have liked to say that you want to add, you can do that. Or and if there are no questions, I suggest we continue with the next talk.
I do not have further comments. What I can say is that I think we are in a steep learning curve. So if I was thinking about this talk and what has been done in the literature, it's really impressive, I think. So when I look at it, then you could say, yes, the glass is half full. We do not understand risk, but it's certainly also half full. It's not half empty. It's also very much half full. We are, increasing understanding in a very rapid way. Also because, there's a lot of research budget available now. Far more projects than
Yes, now I see we have a question and I can read it to you. If exposure levels and exposure sources to nanoplastic is still essentially unknown, how can lifetime accumulation in children and adults be assessed?
I understand the question. So how can we assess exposure if we do not know concentrations? Well, my answer is we cannot. So the lifetime exposure that I was talking about is from a paper where this was not for nanoplastic. This was for microplastic. So this was 1 micron and larger. So that assessment was not for nanoplastic. That was still just untouched in that paper.
Yeah. It's another question for you. Plastic in general contain unknown chemicals. How can this risk of unknown chemicals be assessed?
The risk of unknown chemicals is difficult. So there are several lines of thinking, one of which is still, if you are an analytical chemist, you know that there are a lot of tools available to make unknown chemicals known, just by chemical analysis. There are also other lines of thinking, which is that you would measure more some parameters of chemical presence, or that you would use a response wave. And you could use an assay which provides a response that you know has to come from a certain category of chemicals. So you would not need to know the individual chemical identity, but you could have a view on the overall toxicity relevance of a certain chemical fraction in plastic, and then use that. But at the end of the day, we cannot say that a risk assessment will also always be surrounded by certain assumptions, which leave uncertainties. And then, of course, this is more in the regulatory context that people then use safety factors or assessment factors to just try to cover that remaining aspect of uncertainty, to be on the safe side.
Thank you. It's yet another question for you, and that's what about endogenous biomolecules like steroids and hormones, etc., partitioning into small plastics? Assuming that these plastics have crossed the GI tract and become systemically relevant, can MNPs become a sink for endogenous molecules that then are no longer available to the organism?
In terms of a mechanism potentially be relevant, the question should always be, well, yes, let's consider it and think about it. So when a particle enters a body, a xenobiotic particle, what will that particle do there? Will it dissolve? Will it release some toxic chemicals? Or will it attract certain chemicals that are just part of the natural processes occurring in our body? Now, plastic is hydrophobic in nature, so I would not expect high concentrations of naturally important hydrophobic chemicals to be present. On the other hand, plastic particles have a certain surface, and that surface is not necessarily biocompatible. So it is not unlikely that there could be some interactions in certain parts of the body between a certain surface area composition and biomolecules, which at some point is disturbing. So yeah, that is basically toxicology, and I guess if we keep this question in our mind, maybe one of the later speakers
can touch upon it.
Detail this also.
Yeah. I think actually your time is out, and we should continue with the program if there are no really urgent questions that needs to be posted. So the next speaker is Domagoj Verbos and Severin Koch, and sorry if I pronounced your name wrongly. Domagoj is coming from EFSA and Severin from BFR in Germany, and the title of the talk is Interplay Between Science and Society, the Evolution of Research, Public Disclosure and Societal Concern. So please, you have the floor.
Thank you, Helle. Good morning, and thank you for giving us the opportunity to provide a bit of a societal perspective to the issue of microplastics. Bart already touched upon this in his presentation, and we will be happy to complement this with some further insights from a social science angle. But just maybe first a brief presentation of the speakers. So as you mentioned, my name is Domagoj Verbos. I come from EFSA as a social scientist working in the strategic communication team. And I would just give quickly the floor to Severine to present herself. Severine, please.
Thank you very much, Domagoj. So my name is Severine Koch. Helle pronounced it perfectly fine. And I'm a researcher with a background in psychology. And I've been working at the German Federal Institute for Risk Assessment since 2016 in the Department of Risk Communication. And then I give back to Domagoj. Thank you.
Thank you, Severine. Let's go through the presentation so we can see what we will be talking about today. First of all, we will do a very quick overview of how research on micro and nanoplastic evolved in terms of volume and type. Bart touched upon this a bit. We will expand a bit. Then we will see how and if this translates into interest of people. Do they look for info on these topics? How does media report on microplastics? And finally, we will show data on level of concern of Europeans on microplastics in food and see if we can understand what drives this concern or if further research is needed. And as we already heard, we will have another speaker this afternoon that will go more into the detail of how concerns are formed, Professor Paul, in the exposure session. So, first point to note, and we've seen this already, both microplastics and nanoplastics are topics where research is increasing. Research on microplastics has been around somewhat longer. First paper was published in 1978. Nanoplastics are a bit more recent subject of study. Both experienced quite a sharp increase starting around 2015, and around 85% of research goes to microplastics. So microplastics is a topic that is a bit more explored than nanoplastics. So what is this research about, and which disciplines have explored the issue most? Now, we did a little bit of analysis from Scopus covering around 7,500 research papers. The majority is from the area of environmental science, around 40%. Agriculture, earth and planet sciences, chemistry follow, together accounting for more than 2/3. But what is also interesting, and what interests Severine and myself, the social sciences comes with the contribution of around 130 papers to date. And we know that slowly, this is a phenomenon explored in the realm of psychology, behavioral sciences, just to name some of the disciplines. A recent article in Nature that you might have seen that explores the uncertainty about the risks associated with microplastics among lay and topic experience respondents is a good example. I'm also very happy to see a paper on nanoplastic perception in our poster session. So it seems that social scientists are picking up. So is it the citizen interest and concern in the topic that drives social scientists to increasingly explore the topic? Well, to answer that, we wanted to check a bit if there was an increased interest in micro nanoplastics from the search data from the web and newspapers, and it seems to imply it's a yes. Here we see a Google trend in the web and news search for microplastics in blue and nanoplastics in red, a bit of a dip in the past year due to other public health concerns driving the debate, as we know, but still an overall positive trend, especially web search. And this is interesting because web search is more of a personal thing, we can say. The news search will be a bit driven by the public, by the media, but the web search will be a bit more personal for people's interest. Nanoplastic data is lower because we are using the same scale, but it also works in the direction of increased interest. And then I just wanted to show you a bit the more personal web search, a geographical view of the interest areas. So we see more widespread interest globally in microplastics, dominated by US, Australia, New Zealand, but also well represented in Europe. And for nanoplastics, a bit more localized split, so with Google users from Canada and Spain being most active. Okay, so now we saw a bit on the research and the interest estimates. So how do these combine in the message that the public, that is there in the public, and does that translate into concerns? Well, I don't know if I'm sure you are familiar with the paper from Volker et al. From 2019 on the creation of risks. This was an interesting analysis of how risks are framed in science vis-a-vis how they are framed in the media. And we thought it would be interesting to just show this to compare what we know from science vis-a-vis how we perceive risks. So here you can see when it comes to the studies on microplastics, the vast majority actually deals with hypotheses, so potential risk to be investigated, and only a fourth of studies actually talks about the risk, and most of them from an environmental point of view. So that we would say, is that mirrored in the media analysis? The representation of what is debated publicly, does it mirror what we see in the research papers? Well, that's not necessarily the case, because we see that what the media talks about mostly is the impact of microplastics. And here there was an analysis of 186 media articles in this paper, and basically it showed that the majority actually picked up the risks and impact linked to the human food chain. And remember that the impact of the human food chain would have just been a small portion. If we go back here, so that's in the risk portion, in the 24%, the impact of the human food chain would be an even smaller portion on that. But still, the articles that actually talk about risks, they were the most picked ones, and news articles that were written on microplastics picked that issue up. So the most picked up aspect, despite the quite narrow scientific opus, and I'm unpacking a bit the issue that Bart mentioned on why the concern is high and the level of, let's say, the risk we know is a bit lower. Well, there's the whole social amplification aspect and the media, and how then that translates into interest and concern. I just wanted to, for the couple of examples of media articles, to see what risks they refer to. Fish, the most common example. Actually, fish have somewhat become a symbol of the microplastic discourse. We see it in the existing communication of the topic. But also example of microplastics found in honey and beer, which have been cited in the media. So now the question is, has this transferred into perceived high risks of microplastics? And are citizens concerned about finding these in their food? And this is what we are interested in today from a social science point of view. The recent paper I mentioned from Nature demonstrated a certain level of uncertainty, not only in lay people, but also respondents who study work on the topic of microplastics as a pollutant. And this uncertainty ranges from level of concern about microplastic to concern about existing evidence, etc. But I pass the floor to Severin. She will tell us more on the situation in the EU and Germany when it comes to concern. Over to you, Severin. Thank you.
Thank you very much, Domagoj. Yeah, so from a social science perspective, it's of particular interest whether people are really concerned about microplastics or not. In the 2019 special Eurobarometer on food safety, you see that the level of concern varies considerably between different EU countries, with higher levels of concern, for instance, in Denmark, Sweden, the Netherlands, and Germany. Italy belongs to countries with somewhat lower levels of concern, with 15% of respondents mentioning microplastics in food as one of their top 5 concerns from a list of 15 food safety issues. We have more detailed data from Germany based on the BFR Consumer Monitor. That's a population survey that is conducted twice a year cross-sectionally. And in this graph, you see how the awareness for microplastics in food has developed over the past few years. And as you can see, from 2015 to now, there has been a substantial increase in the awareness. And it's currently about 93% of respondents who are aware of this topic. The Consumer Monitor also covers a range of other health and consumer topics, most of which have lower awareness rates compared to microplastics in food. For instance, 48% of respondents are aware of listeria in food, for instance. A topic that has comparable awareness rates to microplastics in food is antibiotic resistance, shown here in red. And as you can see right now, the awareness rates for these two topics are about the same. But it has been high for the antibiotic resistance for a longer period of time, whereas microplastics in food has experienced an increase over the past few years. Then on the second graph below, we see the levels of concern about microplastics in food and how this has developed over the past few years. And again, we see an increase from 2016 on. And again, this is a comparison with antibiotic resistance. And we see that right now, the concern rates are about the same, with around 60% of respondents being concerned about these two topics. But only microplastics in food has experienced an increase over the past few years. There's some more findings from another BFR survey on microplastics in food that has been conducted in 2018. So this is separate from the consumer monitor. And I think what is the most interesting finding in the context of this talk is that a large percentage of the respondents in this survey spontaneously linked microplastics in food, microplastics to food. So when they were asked, where would you expect to find microplastics? The most often mentioned answer spontaneously was in food. In comparison, only 23% mentioned sea and waters as where they would expect to find microplastics. So it seems that for consumers, they seem to have more of a human health perspective on microplastics rather than a environmental perspective. When it comes to perceived level of risk to human health and the environment, both are seen as high-risk areas, but especially this is the case for the environment. But at the same time, so while people are reporting high levels of perceived risks, they at the same time also say, or the majority of them says, that they feel poorly informed about the possible risks of microplastics. So 51% said that. So these are some preliminary findings from social science studies on perceptions of microplastics. And as we see, there is concern, we know that, but there's not so much known about the underlying reasons for this concern. And this will be the focus of a joint BFR EFSA research project that has started recently. And at this point, I want to acknowledge the work of my colleague Nathalie Berger, who has initiated this project together with my co-presenter Domagoj. And Nathalie is currently with maternity leave. So in this project, we are going to compare the perceptions of microplastics in two countries, Germany and Italy. So we have chosen one country with higher levels of concerns about microplastics and another country with lower levels of concern. The project has two main parts. So in the first one, we are going to do a qualitative analysis with consumer interviews in Italy and Germany. And this is followed up by population-based surveys in these two countries to test the insights that we gained from the interviews. We hope that this project will contribute to the field in different ways. So first of all, on the most basic level, we hope that it will generate knowledge about the underlying reasons for the concern about microplastics. And secondly, we hope that it will provide kind of orientation points for risk assessors and policymakers. And finally, we hope to inform with this project also future risk communication on this topic. This already brings me to the end of this presentation, and we would like to thank all the people involved in this project from BFR, EFSA, and the University of Vienna. And Domagoj mentioned it already, Sabine Pahl is also a speaker at this event, and I have to say that I'm looking very much forward to her presentation. Thank you very much for your attention.
Thank you very much for this nice talk on risk and risk perception. We have one question posted for you, and it's for Domagoj. You point out that Canada and Spain are more interested in nanos. Why you think that the interest between nano and microplastic has a geographical point of view?
Thanks for that question, Helle. We didn't go into that exploration. We simply did a Google trend analysis just to see where the search originates. So we would have to do a bit of a follow-up to check what drove the internet traffic in Spain and the US. I will be happy to do that following the colloquium, and I can provide the answer. But usually what we see is that in this case, whether it's an online discourse or if it's a social media issue where we see peaks in the analysis, that usually means that there was either someone, a high-profile story, a news story, or a high-profile, let's say, influencer on social media that posted something, and that could drive the traffic. So we'll need to see a bit. It could also be that more research, there was more discourse in Canada and Spain on this topic in the scientific area. So I will have to check that and let you know. Thanks.
Okay, thank you. Well, I'm wondering a bit whether this great interest in micro and nanoplastics is because people can easily connect to it. They understand what it is, in contrast to a lot of other chemicals with difficult names that they find hard to relate to. What are your thoughts on that?
Maybe I will let Severin, and then I can complement.
Yeah, I think that's a good point, Helle, and it will definitely contribute to the that it's such a topic with also high awareness, and yeah, it also, as Domagoj mentioned, it has a very high media presence, also because the media will not present topics that people really have difficulty relating to. So this kind of like work goes both ways, I think. That it's a topic people can relate to, and also that this is the reason that the media pick up on it probably more often.
And maybe if I can just add to that, I also think that, we're talking about microplastics, nanoplastics, but they all have the word plastics. And there has been so much discussion about plastics in the past, let's say 10 years, but even more. And we know that when perceptions that are formed, people don't necessarily, differentiate between plastics, microplastic, nanoplastic. You hear the word plastic, you think about the environment effect. And we know that some of these, let's say, biospheric values that people may have where they assess a risk to environment, and it might then translate to perception of risk in the human health area is also true. So at the end of the day, there's also, I suspect there is also a linkage with the overall plastics discourse, not necessarily just the micro and nanoplastics.
That's true. I cannot see any more questions in the chat, so I can just encourage people to pose questions and engage in the discussions. So thanks a lot. Sorry, I think there is another question from you. Yeah, there is another one, so we can go back. We have time for that. Do you think the public perception will influence the ongoing research on micro and nanoplastics? That's an interesting question.
Well, I could say that it should. It should in the sense. When I say should, I think it should inform the questions we are asking, right? If the public perception reveals that people are concerned about a certain aspect, and we have to decide of how, of certain avenues to assessing the risk of micro and nanoplastics, and we know that, approaching the assessment with a certain angle will answer mostly the concerns and the perception that people have, then I guess that should influence, if we are talking about the questions and the direction of research. That's something that Severin mentioned in the last slide as well. We want to do this research on perception that we are doing the study, also to inform the way we risk assessors and policymakers in the future, so that we prioritize a bit the assessment of what scientists know is the area that needs to be assessed, but also from the angle that will address the consumer concerns.
Thank you. Then I think we can move on to the next speak. The next talk is given by Welle van Huysen from DG Sante, the EU Commission. And the title of the talk is Regulatory Needs and Challenges to Inform Fit-for-Purpose Research Activities on Micro and Nanoplastics, including outcomes of the SAM report. So please, Welle, you have the floor.
Thank you, Madam Chair, for this kind introduction. I'm Veerle van Heusden. I work in DG Santé of the European Commission, where I'm a policy officer responsible for contaminants in food. And one of the contaminants in my portfolio are microplastics. At the moment, I cannot see my presentation. There it is. Okay. Yes, I will give you today then a presentation on regulatory needs and challenges to inform fit-for-purpose research activities on micro and nanoplastics, including the outcome of the Scientific Advice Mechanism report. While microplastics pollution has already for quite a while attracted the attention of public and policymakers over the past years. Over the recent years, there were many reports on the widespread occurrence of microplastics in soils, in sediments, in marine environments, in fresh waters, in drinking water, in food, animals and plants. And this has amplified the concerns. It has also created a growing awareness, and with this, there is also a perception of risk, which also relates to the broader debate of plastics pollution. But today, we still have incomplete knowledge on the risks of microplastics to human health. So in my presentation, I will first give you an overview of the outcomes of the Scientific Advice Mechanism report. Then I will explain what the policy options are for protecting public health under the drinking water legislation and the legislation on contaminants in food. I will briefly touch upon the EFSA statement on the presence of micro and nanoplastics in food. And I will end my presentation with explaining to you the information needs for a possible regulation on microplastics in food. The Scientific Advice Mechanism of the European Commission consists out of a group of chief scientific advisors who give independent advice to the Commission to inform policymaking. They make recommendations to improve the interaction between policymaking and scientific advice. In their work, the scientific advisors are supported by SAPEA, that is a consortium of academies which provide advice to the scientific advisors in various fields of science. And then the third component of the Scientific Advice Mechanism is a secretariat, which is located in the European Commission's Research and Innovations Department. So in 2019, SAPEA published a report, A Scientific Perspective on Microplastics in Nature and Society. And in this report, a review is given on the available evidence on micro and nanoplastics from a natural science perspective, from a social and behavioral sciences perspective, and it also, the report deals with regulatory and legislative aspects. So I have included the link to this report in my presentation. Then on the basis of this this a priori, the scientific advice mechanism published in 2019 its report on the environmental and health risks of microplastics pollutions, which is a paper with recommendations for policy regarding micro and nanoplastics, and also the rationale for the recommendations is explained in this report. So below you find also the link to the SAM report. So as regards the occurrence of in ecosystems and in humans, the scientific advisors concluded that there are few studies available on the occurrence in nature of microplastics that have a size lower than 10 to 50 micrometers. Smaller micro and nanoplastics often remain below the limit of detection of the analytical methods. And some studies show increasing concentrations of these plastics with the decreasing size. This means that the actually occurring concentrations might be much higher than those that have been reported until today. As regards the ecological risks of micro and nanoplastics, the advisors concluded that evidence is already available on irreversible long-term ecological risks for some specific coastal waters and sediments. However, if the emissions to the environment continue at the current rate, or if the emissions would increase, the ecological risks could be widespread within a century. As regards the possible health risks, it is concluded that the toxicity and ease with which microplastics cross biological barriers, that this is expected to increase with a decreasing size of the particles. At present, there is no evidence of a widespread risk to humans, but there are significant grounds for concern. So it is concluded that a better understanding is needed on the effects of different concentrations, compositions, sizes, and shapes of micro and nanoplastics on ecosystems and on humans. In the report, some precautionary measures are proposed. The precautionary principle may be invoked when there is an uncertainty on the risk to human health or the environment of a product or a substance. The growing evidence of hazards of micro and nanoplastics, together with their long-term persistence, justifies to take measures to prevent the release and formation of micro and nanoplastics in the environment. These precautionary measures should, amongst others, be to limit unnecessary uses of plastics, to restrict intentional uses of microplastics, to prevent the formation of micro and nanoplastics in the life cycle of plastics, to avoid the release of micro and nanoplastics in the environment, and to try to mitigate and control microplastics from source to sink. Furthermore, experts and the society have to come to a mutual agreement on the risk level and take appropriate responses to it. As plastics play a crucial role in modern life, changing the human behavior will be essential. And values, motivations, incentives, and also supporting measures can help to facilitate pro-environmental changes. Many of these precautionary measures are already included in the EU's strategy for plastics in a circular economy, and so their implementation is ongoing. The aim of the EU plastics strategy is to protect the environment from plastics pollution. The strategy aims to make recycling profitable for business. So by 2030, all plastics on the EU market should be recyclable. It aims to curb plastic waste, for example, by the reduction of single-use plastics, the reduction of the use of plastic fishing gear, Also, a restriction of the intentional use of microplastics under REACH is upcoming. The strategy aims also to take measures to stop littering at sea and to drive investment and innovation to try to minimize plastics waste at the source. The EU strategy has the ambition to initiate in this regard a change across the world. I also included the link to the web page of the EU Plastics Strategy. In the scientific advice mechanism reports, several recommendations are made. The first one is to broaden the policy cover to prevent and reduce microplastics pollution. For this, existing broader legal frameworks can be exploited, such as the Water Framework Directive, air quality legislation, also fertilizers legislation to deal with sewage sludge. And then these general policies can be supplemented with specific measures, which are substance and context specific. And so these specific measures should be science and risk-based, and especially the high emissions and the intrinsically hazardous sources of microplastics should be dealt with. And examples of such measures are, for example, standards for washing machines, special licensing conditions for pellet producers. Furthermore, it's recommended that the knowledge gaps would be filled regarding presence, concentration, and behavior of nanoplastics pollutions, because policymakers have to be equipped with the necessary knowledge to take preventive or risk mitigating measures in case of scientific evidence of human and or ecological health risk. Then the second recommendation is to address the wider socio-economic and trade-off implications of microplastics pollution policy actions, because the preventive measures that are taken, they have to be politically and also economically feasible. Impact assessments of the measures have to be carried out to make sure that these measures have a net positive and sustainable benefit to the society. The measures should be embedded in circular economy logic in order to ensure a better environmental, economical, and social outcome of the plastic system as a whole. And it's considered that the public concerns can help to implement difficult environmental and health protection actions. The third recommendation is to promote global cooperation, high quality scientific exchange, and policy coherence. So the EU aims to promote a global strategy to reduce plastics and microplastics pollutions. It aims to facilitate international and cross-disciplinary cooperation and knowledge sharing. It aims to promote advances in the detection, measurement, and analysis of micro and nanoplastics, and also advances in risk assessment methodologies. Furthermore, it's indicated that it needs to be encouraged that dose response and no effect studies would be carried out. And it should also be encouraged that consensual international definitions would be developed, as well as standards for measurement and monitoring. Now, regulatory measures to protect human health can be taken under the legal frameworks for drinking water and the legal framework for contaminants in food. I will start with the EU Drinking Water Directive. So this directive includes a watch list mechanism to add substances of growing concern to this list in a flexible way, and then these substances will be monitored. The first watch list will be established in 2022. By 2024, the Commission also has to adopt delegated regulations to supplement the Drinking Water Directive. And in one of these delegated regulations, a method has to be adopted to measure microplastics in drinking water, with then a view of including them in the watch list. This watchlist mechanism will enable a follow-up on new knowledge on the relevance for human health of emerging compounds such as microplastics. And by 2029, the Commission has to submit a report to the European Parliament and the Council on the potential threat for microplastics, from microplastics to sources of water intended for human consumption. Then now some information on the regulatory framework on contaminants in food. So the main piece of legislation is Regulation 315 of 93. This regulation indicates that food containing a contaminant in an amount which is unacceptable from a public health viewpoint shall not be placed on the market. The contaminant levels have to be kept as low as can reasonably be achieved by following good practices at all stages. And in order to protect public health, the Commission may, where necessary, establish maximum tolerances for specific contaminants. And this is what's being done under Commission Regulation 1881 of 2006. So under this regulation, maximum levels for very specific contaminants in specific food items are established. These maximum levels can be established when evidence becomes available, which indicates that there is a risk to human health. And then these maximum levels are established on the basis of occurrence data according to the as low as reasonably achievable principle. Furthermore, also some more soft regulatory work is done on contaminants under the form of Commission regulations. For example, to gather occurrence data for specific contaminants, to develop mitigation measures, to also describe, encourage, and in some cases oblige good agricultural, manufacturing, and storage practices. And so in some cases also guideline levels, target levels, and action levels are being set. So while maximum products with the max concentrations of a contaminant exceeding the maximum level cannot be placed on the market, these, for example, guideline levels or softer levels that have to be targeted by the industry. In 2016, EFSA published its statement on the presence of microplastics and nanoplastics in food. And there it concluded that based on a conservative estimate, the presence of microplastics in seafood would have a small effect on the overall exposure of humans to additives or contaminants. However, toxicity and also toxicokinetic data are lacking for both microplastics and nanoplastics in order to allow to carry out a human risk assessment. And then the EFSA statement lists data gaps which need to be filled to allow such a consumer risk assessment to be carried out. So, as I already explained, regulatory measures for micro and nanoplastics in food, so the setting of maximum levels, can only be considered when there is evidence that these plastics could cause a human health risk. And therefore, we need a human health risk assessment, so we need occurrence data. But today, there is a limited availability of such data for food and drinking water, but much more data are needed. Furthermore, an exposure assessment needs to be carried out, and although some rough estimates have been done, so such as a suggested intake of 1 credit card in microplastics a week, we need much more refined estimates. And then there are many question marks as regards the risk to human health. There are possible microbiological risks. There are possible chemical risks due to absorbed chemicals. And then there are possible risks of the plastics themselves. So despite of all the public concerns, it would be preliminary today to regulate micro and nanoplastics in food. So what are the regulatory needs to maybe possibly regulate them in future? Well, the top priority should be to harmonize the definition of micro and nanoplastics, to develop validated analytical and sampling methods, and to also harmonize method requirements. This is needed to ensure that results from different studies can be compared with each other. And then once methods for analysis, harmonized methods are available, then work can be started on toxicological and toxicokinetic studies, on gathering occurrence data, identifying the sources of microplastics in food, and also on the development of good practices to mitigate microplastics in food. Thank you for your kind attention.
Thank you, Eli. For sure, it's a lot of unknowns. So we have some questions from the audience. The first one is from Henriqueta Loro. It says there is an evidence of microplastics in raindrops in USA. They point at the existence of anthropogenic pieces of microplastics, presumably from car tires. Do you consider that the anthropogenic or environmental microplastics can be of more concern, or micro or nanoplastics that are added to products are prevalent?
Well,
Was it clear?
I think that all micro and nanoplastics are from an anthropogenic source. So, but yeah, the Commission is aware also of the problem of the abrasion of tires, and they indeed are an important source of micro and nanoplastics in the environment, and so this should be dealt with. Intentional uses of microplastics and nanoplastics, I'm not really aware of whether there are intentional uses, but you can limit that more easily. But they are a very small fraction, they form a very small fraction of the microplastics in the environment. The majority comes from abrasion and of plastics, deterioration of plastics, pollution in the environment. And once the pollution is in the environment, it's much more difficult than to deal with it.
Thank you. The next one is from Begona Espina. Is easy differentiating between fuel-based plastics and bioplastics for the regulations? Are recycled plastics to be considered for separate regulation?
Yeah, well, there is work going on within the European Commission on plastics recycling. Unfortunately, I'm not an expert in that. I deal with contaminants in food. So as regards plastic recycling, I'm sorry, I cannot give much more information on that. But I will. You can have a look at the website of the EU plastic strategy, and maybe you can find some more information there.
Okay, thank you. And we will hear more about cycling later on, I think. But it's another one. It's from Jane Munke. In the 2019 SAM report mentioned some precautionary measures, including limit unnecessary use of plastics. Is EU Commission working towards identifying what plastic use are essential?
I think, as far as I'm aware, because it's also not my field, but a definition of essential uses of plastics I'm not aware of, but a lot of work has been done already to phase out single-use plastics. And you really see that already now in society. They don't have the plastic bags in the shops anymore, the plastic straws, the ear cotton sticks are now made with paper. So yeah, in that perspective, some advances have been booked.
And I think we have time for a final one. It's also from Jane Munke. If the normal use of plastic food packaging generates micro and nanoplastics, as some few scientific studies have shown, should this be addressed in the EU Commission revision of food contact materials regulation?
Yeah, it's a difficult question. of course, I'm sure it will be discussed when the food contact materials legislation is under discussion, but you cannot forget that plastics also have, in food packaging, have huge benefits, for example, as regards hygiene. Other materials also have their problems. So a thorough risk-benefit evaluation will need to be carried out. You cannot simply just say, okay, we will ban all plastic uses. You have to make sure that you only ban the ones that have, without generating a negative impact on society. Thank you.
Thank you. I'm sure we will continue this discussion also tomorrow. So now it's time for a break. The break will last for 20 minutes, so we will resume again at 11:20. Thank you.